Hello, everybody. My name is Martina Oppliger, and I'm a master's student at Biochemistry and Bioinformatics program of the University of Concepcion, Chile, and I'm part of the team working with Dr. Alexis Salas in the understanding of structure and function relationships of transport systems. Particularly, in this case, I'm working in the understanding of molecular bases that drive substrate selectivity of sugar carriers. Okay, so we all know that sugars such as glucose and related polyols are essential components in the metabolism of all living organisms. The transport through biological membranes is mediated by members of the sugar porter family. This family is composed by homologs of the genes that encode the known GLUTs in humans and animals. To advance in the understanding of the molecular bases that explain the varied selectivity exhibited by these transporters, we carried out the present work. The structural and functional information of various members collected over the years and stored in databases allowed us to evaluate the level of association between the topology of phylogeny and substrate selectivity determined by curation and analysis of functional labels with empirical evidence stored in database, in this particular case in SwissProt database. Residues and regions that are most likely to determine substrate specificities are also identified and characterized by means of conservation, covariation, and supervised-based methods analysis, contrasting our findings with literature. Briefly, we started downloading the representative sequences of the family stored in the transporter classification database. A total of 139 initial sequences were obtained, which were used as input for an homology search against the SwissProt database. Proteins stored in SwissProt not identified by BLAST but associated to the family by the Interpro entry 003663 and or labeled under the DC code of the family in Family and Domain section of SwissProt were also included in the dataset, obtaining a total of 359 non-identical sequences. To eliminate redundancies and fragmentary sequences, pairwise alignments and topology prediction were computed. Details of the steps are present in the document. The filtering process left us with a total of 345 sequences. Subsequently, a refined multiple sequence alignment was obtained. After the elimination of sequences that did not align well in the transmembrane regions, a final dataset of 341 representative sequences of all kingdoms of life was obtained. This MSA was used as input for the rest of the analysis, in this case for the phylogenetic reconstruction, conservation analysis, and statistical coupling analysis. Here we can see the unrooted phylogram of the family and related sequences. Regarding the existence of correlation between the evolutionary relationship of the sequences and their substrate specificity in the global phylogram of the family, we can observe branches that group sequences associated with the transport of a clearly defined type of substrate, such as clades associated with transport of kinnate, maltose, glycerol, and inositol. This group of sequences shares high identity and similarity scores, in coherence with the premise that sequences with identities greater than 40% have a high probability of performing common functions. However, other branches group sequences with border selectivities with differential preference for substrate transport, despite the high values of identity and similarity between the sequences. Examples of these are branches that group fungi and plants family genes. The most important finding associated with the phylogenetic analysis is relative to the relationships between the genes that code for the GLUTs in the different species analyzed. The phylogram, here we can see that the phylogram suggests a new classification for GLUTs, also supported by recent studies and confirmed by the following four groups. GLUTs 10 and 12, one group, GLUT-13, that is grouped with another inositol importers, GLUTs 6 and 8, that groups with threllose transporters, GLUTs 5, 7, 8, and 11, and finally GLUTs 1, 2, 3, 4, and 14. The three analyses suggest that their divergence originated from duplication events prior to the divergence of the taxonomic groups represented in the branches, and to the specific to the Metazoa kingdom. Okay, finally and briefly, with the intention of understanding the basis that explains the difference in the observed cell activities, we perform a conservation and statistical coupling analysis. The conservation analysis identifies the well-known conserved motifs of the family, but also the relative entropy identifies that transmembrane segment number 7 possess a relative high degree of conservation compared to residues with a known role in substrate recognition described in literature in other transmembrane segments, here that are highlighted in green. On the other hand, the statistical coupling analysis shows that the architecture of the family will be made up by a hierarchical decomposition of a single main sector, involving the coupling of residues that directly coordinate substrate with residues located along the transport channel that do not interact directly with the substrates, but that could play a critical role in explaining the differential selectivity and transport dynamic of sugar porters. Finally, the sequence harmony analysis suggests that a relevance of residues located in transmembrane segments number 5, 8, and 10 in the determination of substrate specificities, which are less studied than transmembrane segment number 7 and should be the subject of further analysis. In conclusion, it was possible to advance in the clarification of the evolutionary and functional relationships inherent to the sugar porter family. Despite the advance, there are still no clear patterns that allow us to explain differential selectivities. Future efforts to elucidate the molecular basis of selectivity should continue to be developed.